Well, thank you so much for joining our industry session at the ALD Connect annual meeting. I'm Ben Lenail, and I'm a patient with AMN based in Palo Alto, California, where we are blessed with a beautiful sun here in my home office. And so I wanted to introduce this panel by putting it in context. I'm a founding board member of ALD Connect. We started the organization in 2013. And at that time, we had one company working on our disease, and that was Bluebird Bio, starting to work on their gene therapy for the boys. And we made a point of cultivating a biotech pipeline of therapies that could be applied to the many different phenotypes of our disease. And we were successful in that. We had a vibrant landscape of companies working on our disease. And that means that inevitably, we will have a few setbacks. So Bluebird Bio and Minoryx had challenges during their trials. It is not atypical. Many of these challenges are around, perceived safety issues. And I think we as patients, we are hungry for treatments. We are willing to try therapies. We may have a different assessment of the risk-benefit profile than regulators. And we just testified to the FDA earlier this year and showed the burden of the disease and the hunger for treatments. And so I think it's essential that we continue to make our voice heard, that we continue to be organized, that we continue to tell investors, regulators, clinical sites, and researchers that we are, very, very hungry for therapies. So here we're going to hear from a number of companies that are in trials or about to start human trials, and they will continue to be, the occasional setbacks, but we will overcome them, and we are very hopeful to try these therapies and find ones that are both safe and efficacious for a majority of the patient population. So on that note, I will start with Stephanie Licata from Bluebird Bio, who is going to give us an update on their gene therapy program.
Thanks to come and speak with you. And also, it's been great programming. I've been tuning in all day, and I have to say that hearing the patient panel is incredibly powerful. And so I really thank everybody for sharing their stories. As Ben said, my name is Stephanie Licata. I'm the global medical lead for the cerebral adrenoleukodystrophy program at Bluebird Bio. And there are two important pieces of information that I need to disclose. One is that I'm a full-time employee of Bluebird. I have equity in the company. And also, any of the therapies that I discussed today are investigational, and nothing has been approved by the FDA. As a result, I am restricted by federal regulations regarding what I can say about these products. Next slide, please. Thanks. So I'm only sharing a few slides today. I'm going to give an intro to Bluebird in order to give you some context for what we're doing in gene therapy. And then later, I'll also give you an update on some company news. I'll provide a high-level overview of clinical trial, and I'll share some recently launched educational materials that we've developed in partnership with the ALD community. And I have a note here too as a reminder that if you have specific questions, please reach out to your care team for medical advice. Next slide, please. Next slide. Thanks. So, no, sorry, go back. So Bluebird is pursuing potentially curative gene therapies to give patients and their families more Bluebird days. And if anybody skiing jargon that refers to like fresh powder and perfectly blue skies. And really that is the hope for patients and their families. We have a dedicated focus on severe genetic and research programs for sickle cell disease, beta thalassemia, and importantly for this conversation, cerebral adrenal leukodystrophy. Our therapies are custom designed to address underlying cause of disease, and we've developed in-depth and effective therapies. The expertise really that Bluebird has developed since being founded in 2010. We have the largest and deepest ex vivo gene therapy data set in the world, again, setting a standard for the industry. And then when you couple that to a really people-centric culture in the company that attracts and grows what we always have our bird puns, like a diverse flock of dedicated birds. And this really keeps us driving forward. Next slide, please. So this next slide is an overview of therapeutic approaches, and this encompasses both allogeneic stem cell transplant as well as gene therapy. And this sets the stage for talking about our clinical programs and also really underscores that there's a lot of exciting developments in the broader severe genetic disease community right now. So I think you're very familiar with allogeneic stem cell transplant based on everything I've already heard today in the programming. And this has been used for some time to treat blood cancers as well as severe genetic diseases. The concept is that a person's disease stem cells can be replaced with those of a healthy donor in an effort to treat the underlying disease with the healthy cells. And with gene therapy, we're trying to treat disease by making changes directly to the. So gene therapy has the potential to replace the function of a faulty gene with a healthy copy of the gene, or it can inactivate or silence a gene to interrupt a disease process, or it can introduce a new or modified gene into like certain cells to help treat a disease. And we use the approach of gene addition, and this means that we use an integrating vector, which essentially acts like a delivery truck into the cells, and we do this to insert genes into the genome. But the gene therapy toolbox also includes gene editing, and this allows specific cuts and edits to be made to modify or introduce a new gene to help treat disease. Next slide, please. Our investigational gene therapy drug product candidates. So we're developing Lenail as a potential treatment for cerebral ALD, and we're developing Beticel as a potential treatment for patients who have beta thalassemia and require irregular red blood cell transfusions. And we're developing BB1111, which is a potential treatment for sickle cell disease. And as I mentioned at the beginning, none of these therapies are approved yet by the FDA. Next slide, please. So the clinical development program for Lenail in CALD started a number of years ago with the ALD 102 study, which is complete now, and 32 patients were enrolled, most of whom, after 2 years in ALD 102, transitioned into the long-term follow-up study, which is called LTF 304, for an additional 13 years of follow-up, so that all patients are followed for a total of 15 years. There's an ongoing phase 3 study, which is ALD 104, which is also fully enrolled. And these patients also are set to transition into long-term follow-up 304. So again, all patients are followed for 15 years. Both studies enroll patients with early CALD, and a key difference between the studies was that ALD 102 excluded patients with matched sibling donor, whereas ALD 104 had no such restriction. And more information about all of these trials can be found on clinicaltrials.gov. Next slide, please. So this schematic shows the Lenail treatment process. It is ex vivo, which is Latin for outside the living, meaning that the cells are collected from the patient and transduced outside of the body. So the hematopoietic stem cells need to be collected from the bone marrow, and to do that, they need to be quite literally mobilized from there and collected by a process that's called apheresis. The cells are then shipped to our manufacturing site and selected. We can work with a specific cell type of stem cell. And they're transduced with lenti-D lentiviral vector, which inserts a functional gene into the cell to correct disease. And then the cells are cryopreserved, which is a type of freezing, and they're tested for quality and safety, and then they're shipped to the infusion for stem cell transplant. Now, prior to the infusion, patients undergo myeloablative conditioning, which gets the bone marrow really like ready to receive the cells by making room for them so that the transduced cells can take hold or engraft. And some of the cells, when they engraft, can make their way into the brain and become microglia, which is a type of cell in the brain that's hypothesized to help stabilize the disease when expressing the functional ALD protein. Then, as I mentioned, the patients are followed for a total of 2 years in the feeder studies before entering into the long-term follow-up for an additional 13 years and a total of 15. Next slide, please. So having said all that, I want to shift gears and provide an update on what's been going on recently at Bluebird, because I think this is really the information that the community wanted to hear. And 2021 has been a really significant year for Bluebird. As many of you know, probably we separated into 2 independent companies. So one, which is Bluebird Bio, is focused on severe genetic disease, and the other is 2seventy Bio, that is more focused on oncology. So as part of that process, we're learning, we're growing, and we're trying to understand way to get the therapies to patients. In August, we announced that we decided to concentrate our severe genetic disease focus, our severe genetic disease business on the United States, and we're winding down our independent operations in Europe. And this was in part due to the fact that after more than 2 years of effort, it was clear that the European authorities were not willing to recognize the value of a one-time and potentially curative medicine. In addition, we did have a period during which our sickle cell disease studies were on hold, and like most of the world, we faced delays due to the pandemic. We're a small company, we're an innovative company, and we needed to make choices that will allow us to continue to develop and deliver new gene therapies for patients who need them. And in this case, we had to reach a difficult decision, a difficult conclusion that the best way to continue this crucial innovative work is to focus our efforts and therefore scale back our geographical footprint. And this decision was really difficult for everybody at Bluebird because we believe in these therapies, and we want nothing more than to make them available for patients. And it has been really a difficult time. We wanted to find a partner to assume responsibility for our authorized products in Europe, but despite our best efforts, we have been unable to find that partner. And so
Stephanie, you have 1 minute left.
Okay. We're continuing to investigate ways to make these therapies available to patients outside of the US, but the path forward is unclear. Separately, we announced earlier this year that we received reports of myelodysplastic syndrome, or MDS, which is a type of blood cancer in 2 patients treated with Lenail, which is our investigational gene therapy for early CALD. And the FDA has put the Lenail program on a clinical hold since then. And our hearts go out to these patients. They are continuing to receive the very best care from their physicians. And based on our investigation, we believe that the MDS is likely related to insertion of the lenti-D lentiviral vector. We are continuing to work closely with the FDA in order to, so I will just move on to the next slide then. Quickly, I guess. Right, so just to leave you with switching gears again, highlighting a new resource that we've developed in partnership with the community. This is a newborn screening for ALD advocacy toolkit, the purpose of which is to raise awareness of this life-saving opportunity with newborn screening, and also to raise awareness around the critical importance of early detection and monitoring for disease progression in ALD. This was developed in partnership with ALD Connect and other advocacy organizations, and who are all, hopefully this is fit for purpose, and you can find the resource online as a downloadable PDF at the patient resource section of bluebirdbio.com.
Okay, wonderful. Well, Stephanie, thank you so much.
Thank you. Yeah.
Then we, I want to emphasize that gene therapy will be discussed in more detail tomorrow. There will be a session specifically on gene therapy with Bluebird and SwanBio and others. So now we're going to turn to Marc, who has been gracious enough to join us from his office in Spain, where it's very late. And he will give us an update on the Minoryx program.
Thank you, Ben, and thank you for the opportunity to be here today. It's a pleasure, even if it's late, not that late, it's 9 in the evening, so happy to be here. For those that don't know us, I have a first slide to just introduce who is Minoryx. Maybe not all of you are familiar with us. We're a company from Barcelona. We also have a part of the team in Belgium, in the south of Brussels. And we are a company that I founded now 10 years ago with the ambition to develop drugs in the orphan CNS space. And actually, most of our work is on ALD, which is our lead indication with our molecule. The molecule is called leriglitazone, which is a small molecule, is an oral treatment, and it's what we call best-in-class PPAR gamma agonist. This is a family of molecules that have a lot of potential in CNS, but what has been lacking so far is a PPAR gamma agonist able to reach the CNS in humans at sufficient extent. And this is precisely what we have with our molecule leriglitazone. As I said, ALD is most of our time is on ALD, is our lead indication, but the molecule also have a potential for other indications, including Friedreich's ataxia, among others. To talk about a little bit of the molecule without entering into the details, ALD is really complicated disease. Even if it's a monogenic condition, which may sound simple, the end is very complicated. So there's many different pathways that are affected and that are leading to disease progression. The beauty of PPAR gamma agonism is that through a specific mechanism, we can modulate most of the relevant pathways for disease progression. This includes inflammation, demyelination, neuronal death, and mitochondrial function that are at the core of disease progression, both for AMN and cerebral ALD. We published all this work recently in this nice publication, and there we provide lots of preclinical data showing the potential of the molecule in both forms of the disease. A few years ago, we moved from the phase 1 studies to this pivotal phase 2-3 study in AMN. This was a study that we agreed upon with FDA and EMA about the endpoint design, etc. And in this first study, in the ALD space, we focus on men with AMN. This was a double-blind, placebo-controlled study with 2-to-1 randomization, meaning that for every 2 actives, there was 1 patient with placebo, with once-a-day administration after breakfast, so nothing complicated from that point of view. And we enrolled more than 100 patients, which means that this is the largest trial ever in this form of the disease. And we are very thankful for all the patients, of course, that participated, but also with the clinical sites across Europe and in the US that were involved in this study. As you can see with the names, all the relevant KOLs in the space, or most of them, maybe not all, but most of them have been involved in this study. We treated patients for 2 years, and after this double-blind phase for 2 years, all patients moved to open-label extension. All of those that accepted to do so, or that were willing to do so, that was most of those that finished the 2 years phase of the study. To comment a little bit on the results without entering into all the details, and apologies if this is too simple way to present it, but we wanted to make it simple and easy to follow. In the study, we had different objectives. So we evaluated different aspects of the disease. The most important one, if you want, is myelopathy itself, because this is AMN patients. We want to assess disease progression of the chronic component of the disease. And among the endpoints targeting or looking at myelopathy, we had the primary endpoint that was the 6-minute walk test that measures the distance walked in 6 minutes. We missed this endpoint, so we couldn't see a treatment effect on the 6-minute walk test in this study. At the same time, though, we were able to see pretty clear results in balance. You heard about balance before today. Mr. Finney mentioned balance in his presentation, and this was measured with body sway, which is an objective and quantitative measure of balance. We had several endpoints, several secondary endpoints in the study measuring this balance. and several of them were nominally significant. So that's what means these three marks here. So it's showing a nice result in balance. At the same time, we also got pretty nice results in clinical scales, EDSS and SS-PROM. And all this is related with myelopathy. But another important part of the study was to assess if these AMN patients during the study were developing cerebral progression. As you know, unfortunately, cerebral ALD can occur on pediatric patients, but can also occur in adult patients with AMN. At any time, these patients can develop cerebral ALD. And because the drug in preclinical models has shown nicely that it can modulate or can have an efficacy in both of forms of the disease, we wanted to assess if patients in the study were developing cerebral ALD or not. We look into this radiologically and with central reading, and essentially we saw a very clear result where the molecule or the active arm significantly reduced the progression of cerebral progression radiologically, and also by Lois score compared with placebo. And at the same time, we also got pretty nice results in biomarkers, biochemical biomarkers, markers such as neurofilament light, MMP9 related with blood-brain barrier integrity, interleukins, essentially providing consistency on the global picture because these results are well aligned with the mechanism of action of the molecule. The sites also look from the clinical perspective if patients were developing cerebral ALD or not, and this only occurred in placebo patients, 6 out of 39 for known out of 77 in the active arm. At the study, we also look at other perspectives, so try to get a holistic view on the disease progression. We look at the quality of life, global impressions, so we see trends in there, and we couldn't see an effect on incontinence or erectile function, at least with the outcomes that we used in the study. After the study results were completely evaluated, these are all predefined endpoints. We also did some additional exercise post-hoc. What it means that this is after the breaking the blind. And these were aimed to understand why we missed the primary endpoint. And actually on those post-hoc exercise, what we observe is that the progression in AMN is not linear in ambulation or walking related endpoints. There's an initial phase with a steeper decline, followed by a second phase with a less steep decline. And in this initial phase or early stage patients where the decline is pretty steep, actually we were able to see an impact on the drug, both in 6-minute walk test or in another endpoint that also related with ambulation. So suggesting, of course, this is post-hoc, so it needs to be taken with caution, but suggesting that the drug may also benefit ambulation in the right subpopulation. In terms of safety, the drug was generally well tolerated. We had some adverse events, as any drug I would say. In our case, the most common ones are edema and weight gain. In general, moderate and manageable, and actually reaching a plateau and a stabilization over time. Based on these results, where we think that justify a good benefit risk profile, we went to see regulators in Europe and in the US. In Europe, we had a pre-submission meeting with the European authorities, and after the meeting, we felt—
Marc, you have 2 minutes left.
Okay. We felt encouraged enough to move into marketing authorization application. That's what we are going to be filing next year. In the US, Ben alluded to some setbacks and problems. Actually, we had some transient setback with FDA. We were initially placed on hold on the basis of a failed primary endpoint that was questioning the risk-benefit profile of the molecule, also taking into account the carcinogenic risk of the drug class. We share all the data with FDA, particularly all the data around the cerebral lesion progression. And after this, actually, a couple of days ago, FDA communicated to us that this hold has been lifted for the advanced patients. So all these patients can resume the treatment on the open label extension. So we are very happy about this, of course, and very sorry for all the anxiety that this transient situation may have created in some of you. Additionally, we are also talking with FDA about additional studies that we are going to be preparing in the upcoming months with FDA. The last minute, let me finish with our second study that we have in the ALD space with cerebral ALD patients. In this case, as you know, brain lesions precede clinical symptoms, and after that, there's an acute disease progression. And what we aim to see is that if by using leriglitazone before the transplant is needed, we can delay or even avoid the need of a transplant. This is what we are investigating in Nexus in pediatric patients. This is an open label study. Essentially, we are treating patients in the early stage of the disease, and if we can reach a stabilization of lesion progression or deceleration at 6 months, then we continue treatment. Importantly, we do not interfere with the transplant. If transplant becomes indicated, this is performed at any time. So we do not avoid that if really needed. We are still recruiting in Europe for now, although we have international patients, such as patients from Chile, to give an example. And also the PI is Patty Mussolino from Boston. For sure, you all know her. That's it. I hope that this is on time. I'm happy to answer questions at the right moment.
Wonderful. Thank you so much, Marc. So next, we're going to go to Kevin Finney, who is the CEO of Autobahn Therapeutics in San Diego.
Thank you so much for inviting us. And Ben, it's been a great joy to work with you over the past couple of years. Autobahn is the newest and smallest company amongst this group, so I'll try to be crisp and hopefully you'll hear more from us in the future. The company was formed a little under 2 years ago with a mission to restore hope for people afflicted with CNS disorders. Just a small bit about the team, since we're brand new for most of you. We're in San Diego, California, and while we won't go through the list of the people working for the company on the left, we have a very dedicated group of scientists, including Brian Stearns, who's our Chief Scientific Officer and is an expert in what we will talk to you about a little bit today called remyelination, which we think is a component of potential recovery for AMN patients. And Gadars Davar, who's our neurologist, chief medical officer, and he's had a long career working in neurological diseases, and we're very happy to get him, which is to start up. And the only other comment on the right panel is even though orphan CNS diseases, sometimes it might perceive that it's difficult to get funding and get investors to care about the category, we did a very, very quality investing in the last 2 years. And you'll notice that some big pharma companies, Biogen, Pfizer, and BMS came into the investment with the view that they were interested in both the orphan CNS disease AMN that we're working on, but also the remyelinating program in multiple sclerosis. So I think that's good news for the broader group. Just a small bit about what we do. We are a small molecule, what we call platform company. And very simply put, if I could explain with a laser pointer, in normal states of nature, we all have blood-brain barriers, and the blood-brain barrier is designed to keep things, macromolecules, out of the central nervous system. And so if you take an oral medication and you see down in the bottom panel charged molecules, that oral medication eventually would make it up along the blood-brain barrier, and the CNS is designed to block and keep that out. How can you help improve CNS disease if the brain is blocking and preventing medications from crossing the blood-brain barrier? So our scientists came up with a very clever discovery, and it's effectively you take an oral drug and you make it into a prodrug, which is just a packaged unit. The prodrug technology that we use amidates the drug. It allows the drug here, what we'll see in the next section here, to cross the blood-brain barrier intact through passive diffusion, just meaning it just moves through. And then the prodrug has the ability to be enzymatically cleaved, or the payload is delivered to the brain. So it's an amidating prodrug chemistry. We can apply this to multiple classes of drugs to try to improve the ability to develop drugs to help patients, including AMN. Our pipeline is just emerging, but you will notice the TRB franchise here with ABX002. We've been working very hard for the last several years, and we're just completing our what we call IND enabling work, and we should be going into phase 1 studies in both multiple sclerosis and AMN in the next several months. We're also applying this prodrug technology to improve drugs for PPAR isoforms, multiple drugs in that category. And I think we heard from somebody else today that's showing a lot of promise in that category. and S1P. So sort of the simple point is that we hope you'll hear from us in the future with many more therapeutics that are entering the clinic. I think the group here probably has a very good understanding of the disease of adrenomyeloneuropathy. So we would just like to make a small point today. On the left panel, it describes the disease with the prevalence and the adult form of XALD. I think everybody's become well versed that ALD patients can suffer from ABCD1 gene mutation, resulting in the inability to transport VLCFAs to the peroxisome for degradation. And that can cause what we'll call a VLCFA toxicity. What's a little bit different with what we're doing is we're proposing a dual way that we can help AMN patients. On the one hand, TRBs have been implicated in the ability to stratify and differentiate ABCD2, promote ABCD2, which would be an alternative way of creating transporters to move VLCFAs to the peroxisome for degradation instead of ABCD1. But we also have a pro-remyelination program. So it's a mechanism that promotes a patient's own endogenous, what we'll call OPCs, to actually promote new remyelination. So that's the basis where there could actually be a common thread between diseases, neurodegenerative diseases like multiple sclerosis and AMN. It's important, we believe, to not just treat the underlying causation of disease, but allow the body to actually promote its own remyelination and improve that electrocircuitry. We'll close now with the last slide and just say, so what are we doing? Very early stage San Diego company. We have a very strong investor base that's helping us take this proprietary brain targeting chemistry platform and move it through different candidates to help patients, starting with ABX002, which should be entering the clinic as we finish our IND packaging in the fourth quarter of this year. We also have a great pipeline of new candidates that could help for additional diseases or even additional approaches to treating AMN. We have a great team of committed neurologists, and we have a very strong group of corporate and venture investors that should be able to provide support to advance the program. That's it. Thank you, Ben.
Okay, wonderful. Thank you, Kevin, and thank you for staying on time. So next, we're going to go to David Moller, who is the Chief Medical Officer of Poxel and who's joining us from the Boston area.
It's a pleasure to join you today and give you a quick update on what we're doing at Poxel. I have spoken to this group before, and it's very happy to be back and give you a brief update. And just to remind you, we are a small biotech company. We're based in France, but we have a team in Boston as well as in Tokyo. We've been traditionally focusing on more common metabolic diseases, but now are really getting more interested and focused heavily on two programs in AMN. And this is a quick snapshot of our pipeline. We have the advantage of having a lot of experience in clinical development, including taking a drug all the way through phase 1, 2, and 3, and actually launched now in Japan for diabetes. That's listed at the top of the slide. And then we have two of these molecules that are in mid-stage development that we're initially developing for liver disease. That's called NASH. I'm not going to talk about that today, but what's interesting is that both of these molecules that have different approaches, as I'll briefly describe, are also potential candidates for AMN and ALD. And those molecules are referred to as 770 and 065, and they have, as I mentioned, different approaches associated with them. So these are some of the general features of each of these two molecules. They're both oral agents that can be taken once a day. And we have sort of an advantage in the sense that they've already been studied in healthy subjects in a number of phase 1 clinical studies, but they've also both been studied in large numbers of patients with other diseases like diabetes and liver disease. So with each of these two molecules, we've now exposed over 200 patients and have demonstrated that they're relatively safe and that they have some efficacy in some of the other aspects of the other diseases. The one on the left is focused on working on an enzyme called AMP kinase. I'm not going to go into a lot of detail there, but I'll talk about that briefly on the next slide. The one on the right, which is called 065, is also related to pioglitazone, which has something in common with laraglitazone, the Minoryx molecule. But as I'll briefly mention, I think we have a different take on the way in which we think our molecule is working versus the Minoryx molecule. So that's a bit more expanded upon on this slide. On the left, you can see that for our 770 molecule, we're working through a cellular pathway that's been very well studied and validated in a whole range of different diseases, but in particular has been thought of as a possible way to ameliorate neurodegeneration. And on the right, what we've done is take pioglitazone, which is a marketed drug for diabetes, and modify it in a way that can reduce the PPAR gamma activity, which is that genomic pathway shown here, but preserve activity through some other cellular pathways. I won't go into detail, but these are interesting pathways that just in the last year or two, each of which have been implicated as potential therapies for neurodegeneration, and they have the potential to each affect lipid metabolism or inflammation, and those are components of ALD and AMN. So another way of saying that is that even though our two different molecules are working in different ways, they both have the potential, based on the way we understand the disease today, to affect multiple parameters associated with AMN and ALD by potentially reducing VLCFA levels, but also potentially reducing the toxic effects of VLCFAs that occur in the nervous system. And some of the data that we developed over the course of the last year and a half or so are depicted here. So we have a very similar profile with both molecules, despite the fact that they're working through different mechanisms. And we've done a battery of studies demonstrating that they both have really interesting effects to reduce VLCFA levels in cells from patients, and that's the graph that's shown on the left as an example. And they both have effects that are beneficial in the classical animal model of ALD and AMN, which is the ABCD1 knockout mouse. And there you can see that we have a nice effect to reduce VLCFA in the spinal cord better than pioglitazone, which is the related parent molecule. We also have interesting effects with both molecules. And again, this is just some example data from the mouse model of the disease with respect to histology of the nervous system on the left side of the slide here, where you see that there's a very subtle but interesting phenotype of abnormal neurons in one of the major nerves in the body that's associated with the disease, and that can be largely corrected with both molecules, but we're showing an example with one of the two here. On the right, we also have an effect on the phenotype of these mice in terms of their neurologic function, which isn't as severe as sometimes occurs in people, but is something that can potentially be measured and improved in mice. And that occurs as well with both molecules. So what are we doing with all this information? We have the advantage of having two molecules, and we plan to study both molecules in parallel in two separate clinical trials. So one trial with 770, a second trial with 065. They're both carbon copy clinical trials. They're relatively short 12-week studies with anywhere from, say, 12 to 24 patients with adult AMN, male patients with adult AMN in each study. And the goal of this effort is to really identify which of these two molecules is the preferred molecule that we would want to then take forward into more advanced late-stage development. And we will be measuring pharmacokinetics, we'll be looking at safety, but of course, we'll also be looking at potential biomarkers of the disease, including long-chain fatty acid levels in the blood. And potentially, we might even see effects on other biomarkers, conceivably including neurofilament light chain as well. These two short studies, we call them phase 2A studies, will begin in the early part of next year. And we should have the data by the end of the next year, 2022, and then have the ability to potentially advance into later stage phase 2B3 trials or trial in 2023. That's the game plan. And that's really depicted here, where we intend to complete these two biomarker POC phase 2A studies in the course of 2022. There'll be some time involved, of course, in assimilating the data, discussing with regulators, and then initiating a pivotal study. What I did also want to mention is that we have an open IND for one of these two molecules in the US for AMN, and we have had regulatory conversations that were very favorable within the last month or two for the second molecule. So we should have there's no reason to think we won't have a green light to proceed with both of these studies as planned in early 2022.
Okay, David, 1 minute.
My last slide, just to summarize, we have 2 independent clinical stage molecules that have different mechanisms of action, both oral agents. And the results that we have so far in preclinical models look encouraging, and they indicate that we have the potential to impact the disease in multiple ways with each of these 2 molecules. We'll initiate the 2 parallel clinical studies next year, have the data by the end of next year, hopefully. And there is some potential for expedited clinical development, given that we have largely completed preclinical toxicology, phase 1 studies, et cetera, with these 2 molecules, and might have the opportunity for accelerated approval as well. And we're really delighted to be working with ALD Connect, and are also making a lot of efforts to reach out to other patient advocacy groups that are listed here. So thanks again for the opportunity to update you.
Well, thank you so much, David. Next, we're going to go to Viking. So Joel and Brian, please.
Well, thank you, Ben, and it's really a pleasure for us to be part of this meeting, and we appreciate being included in the meeting. My name is Joel Neutel, and I'm the Director of Research at the Orange County Research Center in Orange County, California. And I have been the principal investigator on numerous of the phase 1 studies with the compound that we're going to be discussing today. I'm also serve as a consultant for Viking Therapeutics, which is a biotech company headquartered in San Diego, California, that specializes in the development of novel drugs for metabolic and endocrine diseases. And today I wanted to discuss with you all a very new and novel drug that I think is going to be very important in the disease that we're discussing, and certainly some of the data that we've seen so far has been very exciting. So let me start off by reminding you all, and some of the stuff I'm sure you're all familiar with, is that thyroid hormone has been something that we studied for many years, and is a hormone that influences almost every cell in the body, every organ system in the body. And we've learned over the years has many beneficial effects, such as impacts on reducing lipids, weight reduction, and really haven't been able to use it much for those conditions, because when overstimulated a little, it may have some negative effects on the cardiovascular system. However, what became apparent in recent years is that the thyroid receptor really has 2 parts, an alpha part and a beta part. And it's the alpha part that almost specifically has the cardiovascular effects, and the beta part that really impacts all the lipid metabolism. And so it seemed many years ago when we started to assess this that it would really be very nice to have a drug that is very selective for just the beta part, so that you could get the benefits of stimulating the beta part without the negative effects of the alpha part on the cardiovascular system. And here you can see finally we have a drug that really is very specific for the beta part of that receptor, and can stimulate it without impacting the cardiovascular system, and allow us to regulate lipid metabolism, reduce LDL cholesterol, triglycerides, and other atherogenic proteins, and also improves metabolic control. And so this was very exciting, because we were now able to really specifically isolate to the part of the receptor that we wanted to stimulate, and now can assess the impacts of that. So I'll go over something that Kevin earlier talked about a little bit, and I think everyone is very familiar with, but I think is important to understand the pharmacology of this drug VK0214 that we're going to be talking about. And that is the fact that we know that XALD is a gene mutation which impacts the ABCD1 gene. And this functions normally to transport very long-chain fatty acids into peroxisomes for degradation. That's what happens normally. In the XALD defect, ABCD1 leads to an accumulation of these very long-chain fatty acids in the tissues, because it's just not functioning to transport those long-chain fatty acids into the peroxisomes. And so you get an accumulation of these very long-chain fatty acids. And we know that that disrupts cell membranes, causes inflammatory demyelination in brain tissue, and motor neuron deterioration. But what was very interesting, and again has become apparent over recent years, that the beta part of the thyroid receptor, the part that I've just talked about, that we've now been able to selectively stimulate with this particular drug, also impacts, or it has an impact on stimulating the expression of compensatory transporters of the same family, the ABCD family, and the subfamily of D2 and D3, which may actually mitigate the very long-chain fatty acid elevation that we see in subjects with XALD. So we know that the gene for adrenoleukodystrophy related protein is now regulated. So we know now that it is regulated by this part of the receptor. And we know that there are alternatives, such as I just mentioned, the D2 and D3 subfamily, that can be induced by stimulating this part of the receptor. And this then may mechanistically compensate for the deficiency in the ABCD1, and therefore may result in correction of the very long-chain fatty acids that we see in patients who have this condition. So here you can see a study looking at this drug that I've been studying now for several years, VK0214, and we can see in the study that it does exactly what we might have expected by stimulating that part of the receptor, and induces ABCD2 expression in human XALD fibroblastic cells or fibroblasts. And you can see very nicely very impressive increases or inductions of this gene in these fibroblasts of patients with this disease. Here is some additional data with the same drug looking at the gene expression in various tissues, and you can see again the same thing, that compared to placebo or vehicle, both in the liver and in the brain, this drug, VK0214, significantly increases the ABCD2 gene expression in these key tissues. So we know that this is perhaps a compensatory pathway that we may have, and we now see from these studies that this has been well stimulated and induced in the various tissues and in the fibroblast cells. We also looked at the ABCD1 knockout model, and I think you all know well that this is the model that we have that best mimics what occurs in subjects with human subjects with XALD. And you can see from this data that when treated with this drug, VK0214, that there were quite significant, in fact, highly significant reductions in the plasma levels of the very long-chain fatty acids, and you can see all of them shown here on this graph. And when we look at the same kind of thing in the various tissues, the liver, the spinal cord, and then in the brain for two of the very long-chain fatty acids, again, you can see the same thing, that treated in this model, when treated with this drug VK0214, they had significant reductions in all of these various tissues of the long-chain fatty acids, confirming, I think, that by stimulating this part of the thyroid receptor, you are having significant impacts on long-chain fatty acids through induction of the ABCD2 gene.
Okay, and Joel, we have one more minute.
Okay, so just to show you some of the data that we did in human subjects, these are the studies that are in our phase 1 unit, and you can see in treating human subjects, these were normal volunteers who had slightly elevated LDLs, but otherwise normal. You could see we showed significant reductions in these subjects in LDL cholesterol. We also showed very significant reductions in triglyceride levels. You can see up to 40%. And in the same subjects, we also demonstrated some reductions in very long-chain fatty acids, even although they were normal to start out with because these were healthy volunteers. So you can see promising lipid reductions, very safe drug, excellent tolerability, pharmacokinetics suggesting a once-a-day regimen. And so as a consequence of this, we've just recently started and randomized the first few subjects with AML. And here's the study that we're doing. These are adult males with AML, and you can see that they will be randomized to either placebo, 20, 40, or a higher dose of VK0214 in a double-blind fashion, treated for 28 days with a 7-day follow-up. And we're going to be looking at safety, tolerability, and changes in very long-chain fatty acids in these male subjects. So very exciting study underway already in the US. Here's the clinicaltrials.gov identifier for those that are interested, and you'll be happy to know that we have now randomized the first few subjects in the study.
Okay,
so
wonderful.
Thank you so much, Ben. Appreciate the time and looking forward to taking any questions if there are any.
Thank you, Joel. And then we're going to go to Asif, and we're going to extend this session by about 3 minutes, Asif, so you don't feel like you're completely robbed.
Hello, everyone. I want to thank Ben, Kelly, and rest of the ALD Connect team for the organization of such an informative meeting. And thank you for inviting me to this session. It's a true pleasure to speak with you. Before I start, I also want to congratulate Marc and his Minoryx team for resumption of AMN trial. It's a great success, good hope. Next slide. I'm Asif Paker. I work at SwanBio, a small biotech company in Philadelphia, and we are developing AAV9 gene therapy for AMN. Today, first I want to give you a short introduction to gene therapy and how gene therapy approach for AMN is different than the gene therapy approach for inflammatory cerebral phenotype. And then I'll share the findings of animal studies depicting the efficacy of AAV9 approach. Genes are made up of DNA, which are blueprints to build proteins that make our body work. For example, ABCD1 protein is built by the gene affected in ALD, and this protein helps break down of very long chain fatty acids. Gene therapy introduces a working version of faulty ABCD1 gene in the body. This is done by using a vector, which is often derived from a virus, but the viral genes have been removed, and only the therapeutic genes are delivered into the cell. The modified cells then produce the protein that was missing or defective prior to the treatment. Now, the vector can be delivered outside the body. That's ex vivo approach that's shown on the right side of this slide, or the vectors can be injected into the body directly, in vivo treatment, and shown on the left side of this slide. The ex vivo approach is used to treat the cerebral form. The Bluebird Bio's gene therapy program, as Stephanie reviewed it with you today. Here, I'll not go into that detail, but blood cells are removed from via blood draw. They are modified using a vector to deliver a working copy of the faulty ABCD1 gene, and then patient undergo a Moller ablative therapy to make a room for those modified cells. And this is ex vivo gene therapy, meaning cells are removed from the patient and then altered and then infused back. This approach is not appropriate for AMN. Therefore, we are working on an in vivo treatment for the AMN. That's the left side of this slide. AAV9 vector carrying the working copy of ABCD1 gene is injected directly in the CSF, the fluid that surrounds the spinal cord and brain. With this injection, AAV9 vector will deliver the functional copy of the gene to the cells of spinal cord. This will provide cells the opportunity to produce healthy protein to slow or stop the AMN disease. With this approach, we do not have to collect stem cells or use myeloablative chemotherapy. It's a direct injection of drug into the CSF or cerebrospinal fluid. With cell delivery, each patient is treated with a unique product each time. Each patient's treatment is developed by a multi-step process, as I described above. Can you go to the next slide, please? Here, you see a AAV9 vector carrying or
yeah, Asif, unfortunately, I'm being told that we need to head over to the awards ceremony. So basically, knowing that you'll be into the gene therapy session tomorrow, maybe if you can wrap it up in about 1 minute.
Okay, can you go to the next slide?
Sorry. Yeah.
No problem, Ben. Let's do it. So one quick one. We showed in mice that this vector can deliver the gene and can reduce the very long chain fatty acids. That's good approach. Next, and then next slide. So here are showing the very long chain fatty acids. Keep going. The one message that I want to give is that in addition to this gene therapy, we're working with Autobahn to develop a patient-reported outcome measure. This is with the help of University of Rochester. This is a new outcome measure that is specific to AMN, and all AMN men and women can participate. More patients, more men and women participate, the better it would be. So I will ask, and ALD Connect is working with us, with ULF and other foundations. If you haven't filled that survey yet, please go ahead, do it, forward it to your friends and families who can participate in there. Thank you, Ben, and thank you all.
Wonderful. Well, thank you to all the panelists. Thank you for the audience, and we should head over to the awards ceremony. Thank you so much. Take good care.
Thank you.